Good morning, everybody. Thank you very much for coming here. So it's my pleasure to introduce Dr. Tina Sorensen Dalgaard. She is an associate professor attached to the Department of Animal and Veterinary Sciences at Aarhus University, Denmark. She has been working with veterinary immunology, especially in the Asian immune system, for 20 years or so, even more than that probably. She obtained her PhD from University of Southern Denmark in veterinary immunology, and then worked in various positions as postdoctoral researcher and a project leader after her PhD as well. Her research focuses on experimental models of infectious diseases for evaluation of natural immunity as well as vaccine-induced protection, with a special interest in cell-mediated adaptive immune response. Moreover, her work comprises studies of immunomodulatory interventions to improve disease control, but also includes research to support understanding of the genetic background for natural disease resistance and optimal immunocompetence, as well as biomarkers for general robustness and resilience. So she has authored over 80 peer-reviewed scientific publications. And I found yesterday that she's currently functioning as an associate editor for Journal Veterinary Research. Yeah, because I was just looking for something to write. Yeah, so today we are honored to have her here with us in Miami, and she'll be delivering a talk on host-pathogen interaction in Ascaridia galli, infected chicken, with some, maybe I hope she may shed some light on based technologies as well, because we have been working with in a little way of developing some intervention using EVs. So, thank you very much.
Thank you very much, Shwanga, for this kind introduction. I'm very happy and pleased to be here. And today, the title of today's talk, as Shwanga presented, is Host-pathogen interactions in Ascaridia galli infected chickens. And this will be a special talk because it has actually been 10 years since we really worked in this research area. So what I present to you today is like really old data, probably techniques you don't even recognize, some of the young people, like old-fashioned RNA isolation and northern blots, histology, really nothing fancy, but I expect a bunch of really good ideas and questions for our future research plans from you guys. But hopefully it will be easy to follow, as there is no omics, there is no pathway analysis, there's nothing fancy in it. We're just going to discuss some of the basic stuff that we know about this parasite. So let's dive in, and I just wanted to show you a picture of where I come from. Aarhus University is obviously located in Aarhus, the second biggest city in Denmark. But the Department of Animal and Veterinary Sciences is actually located way out in the countryside in the middle of the peninsula. And we are quite isolated, as you can see. It gives really nice opportunities to have livestock of different kinds, but it makes it a little difficult to attract students and interact with some of the other research institutes. But I've been working here for more than 20 years, focusing on immunology and vaccinology. 10 years back, I also worked in parasitology, and we're going to actually open this research area again. And sometimes I pretend I know a little bit about veterinary medicine, but really I'm not a vet, so bear with me. My favorite species would be the chicken, and that's where my expertise lies. I also work a little bit with pigs and have worked with mink in previous projects, but really avian immunology is my core expertise. So today we're going to talk about intestinal worms. And is this even interesting in modern poultry production? You would maybe think back to way back in evolution, where the jungle fowl was running around in the forest, and they probably had worms. probably the avian immune system evolved to be able to cope with worm infections. So it maybe shouldn't be that much of a problem. But it actually is. Worms are actually an increasing problem in commercial protein production. And we have disease-causing nematodes and cestodes. Today we are going to talk about the nematodes, the roundworms. And down here I have listed three of our favorite ones, the more prevalent ones. This is Heterarchis gallinarum and various capillary species. It's actually a combination of many species that we have in this category. And then the main focus of today, which is Ascaridia galli, the large roundworm. So what happens to the chickens when they're infected with the large roundworm? Many egg-producing farmers complain that they see a drop in egg production really, really quickly, even before they realize they have an infection. But depending on the severity of the infection, there can also be something with the body weight and the mortality. And there's also something about reduced animal welfare. They get more aggressive and peck on each other. And obviously there can be damages to the intestinal lining. And there is also at least one paper claiming that it limits absorption of nutrients and it sort of tears the energy reserves from the liver. I will show you some data on what happens if you try to vaccinate against viruses in parasitized animals. We will get back to that. And also we have this problem that when they run around with a lot of worms in their intestine, they are much more susceptible to secondary bacterial infections. So a really nice worm model to study and to try and understand what is going on in relation to host-pathogen interaction. And what does it look like globally? What I show you here is a really nice meta-analysis that was published a few years back. And on the map over here, you can see that they have compiled papers from various countries throughout the world. There are some countries totally missing in this meta-analysis, but anyway, it's the biggest analysis we have currently of the worldwide prevalence. And you see here on the prevalence chart that we have our three favorite roundworm species. And they are actually identified in the meta-analysis more than 30 different common species. But Ascaridia galli and Heterarchis gallinarum and also the capillary species, both among them, with quite high prevalences. Also in the capillary species, but you see the average is really, really low. But in some papers, there were like recorded prevalences of up to 90%. What was really interesting in the paper was that they found that if chickens were kept in backyard and free-range systems, they had a much higher prevalence than if they were housed in cages. And what I find really interesting is that the prevalence over time, it was actually a compilation of papers from 1942 to 2019. So over time, they were able to show that the prevalence were actually decreasing in developed countries, whereas in developing countries, whereas it increased in the developed world. So how come this difference? And part of the explanation is, of course, this. We do not really produce that many eggs from caged chickens any longer in the EU. We had, way back in 2019, we had a ban on the old-fashioned battery cages, and this was followed up by a ban of enriched cages here quite recently. And this is why we see more and more alternative non-cage systems, and some of them even have outdoor access. And obviously, this can create a problem in relation to parasite infections, as the parasite eggs accumulate and develop in litter and soil. And some of the parasite eggs are actually also really, really stable in the environment for many years. So, and sanitation is much more difficult when you have outdoor chickens. But of course, that's looking from the EU perspective. As you see on this map, it is really within the European Union, partly also in North America and in Australia, that we see a lot of cage-free production of table eggs. In the rest of the world, we still use cages, and obviously they are not so interested in understanding host pathogen interactions of the roundworm because they don't see the problem yet. What I show you down here is a quite old prevalence study from Sweden with the Ascaridia galli prevalence. But again, we see here that in the cages, the prevalence is really, really low. Whereas if you go to free-range organic hens, 77% of the flocks were actually infected in this old study. And when we go out and look at organic chickens in Denmark today, we see very close to 100% prevalence. We can always buy organic chickens that harbor the roundworm in the intestine when we go out and buy commercial ones. A more recent study from our colleagues in Austria also shows that within these alternative production systems, the organic ones are really the ones struggling the most because they have more access to the outdoors. But you also see a problem in indoor barn systems where the chickens are kept on the floor. And you also see S2 here and S3 are two different time points in the production period. S2 being at the peak of egg production, around 38 weeks of age, and S3 later on, 74 weeks of age when they're taken out of production again. We're actually trying to extend the period to 80, 90, 100 weeks, but that's a different area. But you very clearly see that the organic farmers have more problems. And this is not prevalence, this is fecal egg counts. So this has got more to do with the parasite load that the organic chickens not only have a higher prevalence, but they also harbor more worms per animal. So as I started to explain to you is that we have really not worked on this pathogen for many years. 10 years back, we had a really lovely vaccine project. We were quite naive in thinking how difficult can it be to develop a vaccine? And it is actually really, really difficult. I will come back to why. And obviously, we did not manage to come up with a good vaccine candidate. But in this old vaccine project, we were able to establish an infection model and some immunological tools, and they have just been sitting in the computer for many years because there was a lack of funding. But hopefully, we are now ready to open up this area again, also in collaboration with Suranga and Ederesa, where we are going to study something about EV immunomodulation, but also basic immunology in this infection model. We have had in recent years a few small projects on botanicals, especially for these organic producers. It is quite attractive if you can find a particular plant that you can add to the chicken feed and then it has an antiparasitic effect. It is really, really difficult to identify and to get into the feed in high enough concentration that it actually has an impact. And these projects have not really been useful for us when we wanted to study immune responses and protective immunity, because many of these trials were just comparing normal feed to various feed interventions. So we did not have an uninfected group without parasites. But hopefully from now on, we will do great things in collaboration with Ranga and Ederesa from Tartu here. So let's take a closer look at the roundworm. It is a really big pathogen. Some of the females are actually up to 13, 14 centimeter long. You are not in doubt when you open the intestine and see the worms. The eggs are very, very small and very, very resistant, and they resemble the heterogynous gallinarum eggs. So one has to be a little careful when one does the fecal egg counts, because it can be a little difficult to see the difference. But looking for adult worms, you're not in doubt. These cannot be mistaken with anything else. In the next slide, I will come back to the infection route. And in this slide, I just want to point out what the Danish egg producers have been complaining about lately is when the nematodes actually go to the table eggs. We have like every second year, there's a newspaper article with a picture like this, and it's a big news story that one consumer found a worm in their morning egg, and it happens really, really rarely, but it gets a lot of publicity, the one egg they found per year with this little worm inside. If we look at the life cycle of the nematode, it is so that the chickens ingest eggs from the environment with infected larvae. The larvae hatch in the upper part of the intestinal tract. And a few weeks after infection, there is a tissue-associated phase where the little larvae are actually embedded in the tissue. It's a real histotropic phase, and this is really the only time point where the larvae are in direct contact with the immune system of the hens. After this phase, the larvae returns into the lumen and adult worms develop. And then after 5 to 6 weeks, we see fecal excretion of parasite eggs, and so the cycle continues. And about prevention, it is easier within barns to do nice sanitation, disinfection, burning floors, whereas the outdoor areas are much more difficult. There have been projects with different fungi being able to destroy the eggs, but it's really not that easy, and you can't sort of do any burning of the pasture between different chicken runs. So some farmers use pasture rotation to get rid of the problem, but it's not easy at all. And obviously clean feed and water and normal biosecurity is really important. But we also have a few anthelmintic compounds that can be used. We've got flubendazole and benbendazoles, and they can be used within the EU to sort of lower the parasite load if the farmer feels that now it gets too extended, too severe. The recent legislation says that there's a withdrawal time for organic eggs. So the organic farmers cannot sell their eggs as labeled organic if they use the drugs. And that creates really problems in their production and in their revenue. And obviously, there is a general consideration that we shouldn't use too many drugs because of the risk of resistant development. So I think it's a really nice time to start projects in this research area again, to look at alternative antiparasitic compounds, but also to look to the vaccine development once more. So how do we do that? This is a really nice close-up picture of the worms. And you see we're not operating in the nano and the micro scale as we do with viruses and bacteria and fungi. And it makes everything much more complicated. The physiological and immunological mechanisms are quite complex, depending also on the life stages of the worms. And it really is not difficult it really is difficult to understand the host pathogen interactions in this particular infection model. We have talked a little bit about some of the clinical symptoms, but what I want to dive into now is what we know about the immune responses. And some of you may have studied a little bit of immunology, and you know that from the textbooks, Th2 immune responses are really important in worm infections. And we sort of think we see a Th2 response. I will come back to that and show you the few parameters we've measured. But for sure, it is not a protective immune response. The chickens cannot normally get rid of the infection naturally. I will also come back to this experiment we did where we studied how egg-producing hens responded to viral vaccines if they were infected with the worms. And I will also show you a little bit we did way back, looking at the specific immunoglobulins in the blood to see that there was absolutely no correlation. They play seroconverted, but it had absolutely nothing to do with worm load. And we have not been able to find any relevance for maternal immunity in this type of infection. And I also have a longitudinal study to show you where we can sort of see that there are fluctuations in worm load over several weeks up to a year, but we don't quite know how a potential self-cure mechanism is the mechanistics behind it. But firstly, I want to explain to you the infection model that we have developed. It is running in maybe a handful of labs in Europe, and it's basically the same infection model. You go out and you buy some organic chickens, and you find some Ascaridia galli worms, and then you retrieve the eggs from the uteri of these worms using a microscope, and then you embryonate the eggs in vitro and wait for some weeks before you're ready to infect your chickens. Then we administer eggs orally and wait for the infection to establish. It really is quite simple. Everybody can do it, but when you look closer, it is definitely not an easy infection model. There is something about dosages. Probably in nature, the dose is not that high. When they excrete parasite eggs in the feces, it's probably not a very high load that the neighbor chicken picks up from the ground. And we have tried different doses for the experimental in vivo experiments. And it seems that the establishment rate goes down the more eggs you put into the animals. And also, if we use a lot of eggs, it seems that more male worms develop than female worms. So there are some interactions we don't quite understand. Also, to resemble what goes on in production, we have sometimes bought commercial hens and then started by deworming them. But during the years, it has been established that this deworming actually makes them more susceptible to reinfection. So the question is if it's a really good infection model, if you start out with already parasitized animals, deworm them and reinfect them. And we have also seen huge differences between genetic breeds and different ages of infections. And I will come back to that a little later. And also in commercial poultry production, we seldom see Ascaridia galli infections as a mono-infection. Usually it's actually in combination with Hydragis gallinam or Cavalaria species. And there's also something about if you give the eggs orally, or if you let the hens pick the eggs from the ground, it also gives a different response. And always, no matter how much we really try to control everything, we see large individual variation between the infected animals. And then there's sort of a balance. If we use a high infection dose, we really get a really nice humoral immune response. But the establishment rate, as I mentioned, is not that particular great. So it's always a balance what you want to study, and you can sort of treat the infection model to boost whatever you're looking at. Question is how it translates into the natural infection. So let's talk a little bit about basic immunology. And way back, we studied a little bit of adaptive immunity towards the Ascaridia infection. And we started out by making a really, really simple ELISA where we used crude antigen from adult worms. We simply mashed them and made a protein soup from the worms and used that as the antigen in an ELISA where we could test serum or bile or whatever we wanted to test, and then use secondary antibodies directed against IgY, IgA, or IgM chicken immunoglobulins. And this gave us quite a nice result in the old days, but we never got around to use other antigens. Obviously, it can be really interesting to also create antigen from larvae or even from recombinant proteins, but that will probably come in the future. But I just wanted to show you what it could look like. This is one of these feed intervention experiments. So there are 4 groups, and that's not really relevant for this presentation. So don't focus on the 4 different groups here. But what you see on the y-axis here is serum immunoglobulins, and sometimes we call it IgG, but it is actually called IgY in the chicken. But these are Ascaridia galli specific antibodies measured with this ELISA technique. And you see here a quite nice peak 2 weeks after infection, a decline again, and then a slow increase up to 14 weeks where we ended this particular experiment. And these were old hens from our own organic farm. So they were already infected when we started the experiment. So this was one of these experiments where we started by deworming and then reinfecting them. And if you pay attention to these little color codes up here, they translate into the life cycle of the parasite. And what you can see is that the 2 weeks peak here is actually something in relation to this histotrophic phase where the larvae are really in contact with the immune system. So that was quite interesting for us to see, and we have repeated in other projects. Quite often we see this peak at 2 weeks of age when we use hens, introduction like adult hens in the experiments. But if we use younger animals, naive animals, we don't necessarily see this really early increase in antibody titers. And I want to show you some of the experiments that we've done with young chickens. And for these trials, we have been using our own inbred lines. I don't know how many of you are familiar with chicken immunology, but the chicken major histocompatibility complex is located on chromosome 16 in the B locus. And we have known for decades that this is a really important genetic region that determines resistance and susceptibility to a bunch of bacterial and viral and parasitic diseases. It's actually quite unique in the chicken. The chicken MHC is smaller and more compact than, for instance, the human MHC, and it translates into remarkable differences in resistance and susceptibility to disease. And we have got a number of lines. We have had to let go of some of them, but we still have the yellow lines present at our institute. So we keep them and we breed them and we keep them with a fixed MHC haplotype. So we can go and pick that, for instance, if we study Marek's disease virus, we would go and pick B19, which is highly susceptible, and B21, which is highly resistant. And then we can infect them and study the immune parameters and how the response differs in the different chicken lines. So we really like to use this. It is by no means like using inbred mouse lines. I don't know if one of you have been doing that, but they're much more homogeneous genetically speaking. It is by no means the same, but it still narrows down the variation a little bit. if you can only use animals of the same MHC haplotype. But what I show you here are two experiments where we have in total tested 10 of these lines. Some of them are not actually MHC fixed, but that's a different story. And what you see here is the egg per gram feces parameter, where we have counted Ascaridia galli eggs in fecal droppings from the chickens here, and they were only 4 weeks of age when we infected them. And you see here variation between the different breeds, especially in experiment 2 here, where we've got massive differences in the parasite load of the different lines. And in the lower part here, you see the corresponding immunoglobulin titers in peripheral blood. And as you see, there is also quite some variation in these, and it takes a little while before the chickens actually seroconvert. It's not until week 7, 8 that we even see any antibodies in the blood. And I just want to show you the example with these two lines. These are actually recombinant lines harboring both the B15 and the B21 haplotype. But as you can see, R4 is almost resistant to the infection, whereas R5 is very, very susceptible. It's actually one of the most susceptible lines we have at the Institute. And if you then look at the serology, you see that they basically have the same humoral response, and there's absolutely nothing to be concluded from looking at the serum titers. Over here, we've also measured IgA in bile, and the variation is less profound in bile. This is maybe only for the ones really interested in chicken MHC, but just to show you that these R4 and R5 lines are corresponding to B15 in one region and B21 in another region. It is so that the chicken MHC is comprised of region encoding genes for classical MHC molecules, MHC class 1 and MHC class 2. And in the chicken, we also have some MHC molecules called BG molecules. We haven't studied them in all details, but the classical MHC class 1 and class 2 molecules are the ones located over here. These two regions are separated by a genetic region where we have some C-type lectin genes located. And as you can see, the R4 and R5 are totally identical in this particular region. So if nothing else, we have sort of showed that this particular region is probably not involved with this massive difference in susceptibility and resistance between these two lines. And this is another study where we use this highly susceptible R5 line, comparing it to the pretty resistant B14. If you look at the X per gram feces curve over here, you see that it's a very modest response in B14, whereas the R5 have a massive infection. And this is a longitudinal study where we've looked at the chickens. These were adult chickens for up to 80 weeks. And we see fluctuations throughout the year. It could be seasonal, but it could also have something to do with immunity, with self-cure mechanism. We don't know all the details why this is that we see this fluctuation. But as you see, looking at the serum specific immunoglobulin titers, it doesn't really correspond to the fluctuations we see over here. And the conclusion is really that although they seroconvert, they have systemic antibodies, it does not prevent this reinfection we see later on. So those were the systemic antibodies. How about T cells? This is my favorite subject. love avian T cells, and I used every opportunity I have to try to study them. And this is one of the really old experiments where we had young birds infected with Ascaridia galli, and we were sort of trying to figure out if we in blood samples or in spleen samples could find any trace of any Ascaridia galli specific T cells. And we have this very simple method of doing it using flow cytometry, where we take the cells and we re-stimulate them in vitro with the antigen in question, and then they're supposed to proliferate or start to do cytokines or whatever they are supposed to do. What you see here are flow plots of live cells and singlets, and here we have stains for T cells. And then we simply look at the size and the granularity of the T cells. And in this gate here, we see a lymphoblast, which is really a proxy of proliferating T cells. So re-stimulating cells from peripheral blood, we did not see any difference between control and infected chickens. We were not able to identify any parasite-specific T cells in the blood. Maybe in the spleen with a high, this is a low antigen concentration and a high antigen concentration, see a little trace of maybe an increased response in the spleen. But what was quite interesting was that the positive control was highly different between the two groups of animals. That's a mitogen con A, but it doesn't really translate into us being able to say that this group definitely has some antigen-specific T cells. So really what is maybe more interesting is to look locally in the immune tissue, in the intestine, not in systemic organs or in blood. And from mammals, we know a lot. We know plenty of things about the immune response to helminth. We know we have things we can build our research questions around. We've got the Th2 cytokines, we've got something about eosinophils and basophils, we've got some early alarmins, we've got something about regulatory T cells, we've got M2 macrophages. So plenty of things that we can ask ourselves, does this actually happen in the same way in the chicken? And I just wanted to show you this one. This is from a PhD defense more than 10 years ago. You don't have to look at the details here. Basically, it shows the same as in the previous plot. But our PhD student showed this at her defense, and then she had crossed over things we don't have in the chicken, and she was frustrated about how difficult it actually was to go by the textbook and take one research question at a time. So still to this day, I'm not quite sure we have these alarming genes in the chicken. The chicken genome has been really, really awful to annotate. It's only recently we found the TNF-alpha gene, for instance. It's a very GC-rich genome, so we've had plenty of trouble finding genes. So maybe they're there, we just haven't found them all yet. And in the early days, we even thought IL-5 was a pseudogene. There's something about eosinophils in the chicken. The numbers appear to be low, but we have these heterophil cells that are maybe a neutrophil equivalent, and they are really present in high numbers. But the major thing that we are complaining about is really that IgE does not exist in the chicken. And IgE is such a profound element of helminth immune responses. So that's really frustrating that we do not have IgE. And there's also some differences about the functional development of goblet cells. So as you can see, it's not an easy task just to translate directly from the textbook and see if the same thing holds true in the chicken. So first, really quickly about eosinophils and mast cells. I will not spend too much time off it, but this experiment, looking in blood again, they saw maybe numerical increases in these two cell subsets, but nothing statistically significant. It was not until actually a Danish group looked into the tissue This is like from the genome of infected animals, where they were actually able to see that, yes, the infected animals have increased numbers of both eosinophils and mast cells. And here's another study where they show the same, that mast cell numbers are actually increased in infected chickens. But we still have a lot of things, and this was in particular the 2 weeks post-infection time point, where the larvae are embedded in the tissue. But we still have plenty of research questions related to this pathway, and nobody has looked into the details yet. So more work to do in this department. But then, how about the Th2 cytokines? Those are really, really important. We know that from mammalian literature. This is a slide. We had a visit from the late Pete Kaiser from the Roslin Institute. He's a very well-known avian immunologist. He came over to Denmark 10 years ago, more than 10 years ago, to teach us what avian Th2 responses looks like. But he was also frustrated. He had exactly identified the problems that we also struggled with. And the original hypothesis was actually, oh, do they even have Th2 cytokines, the chickens, or do they have a reduced number of Th2 cytokines? And that's a very fair question. There's a really, really old paper stating that indeed they do have both Th1 and Th2 cells, but the proof is not all that extensive. It's like really old fashioned northern blots looking at mRNA from spleen and intestinal tissue. And fair enough, you do see some expression 2 weeks after infection with Ascaridia galli of the Th2 cytokines IL-4 and IL-13. But it's the go-to. It's still referenced a lot. This one paper we have stating that the Th2 polarization is exactly as in mammals, also in the chicken. So we need to do better. And now we have the chicken genome, and what we have found out is that some of the TLRs are missing, and there are some additional ones, and the pattern recognition receptor repertoire is indeed different. And some of the cytokines might also be different in the chicken genome. But luckily, it appears that the Th2 cytokine cluster here is relatively conserved as compared to the human cluster. So maybe it's not all that bad. I'll drop that one. So some have been looking at intestinal tissue. Can we then, in the tissue, as in this Th1, Th2 paper, look at the kinetics of the infection, and at different time points, can we identify some Th2 cytokines in the tissue by bulk qPCR, very basic analysis? And yes, it appears that at 2, 3, and 6 weeks post-infection, especially at this 2-week time point, a little bit at 3 weeks post-infection, there is an increase in IL-4 and IL-13 locally in the jejunal tissue. So far, so good. We have also shown this, if you look over here, the 2 weeks time point, an increased expression of IL-4 and IL-13. And this was actually the trial I was alluding to in one of the first slides, where we had a hypothesis about Ascaridia galli infection might jeopardize the response to a viral vaccine. I don't have time to go into the details, but the conclusion was that it is bad to have an Ascaridia galli infection. It jeopardizes both the humoral and the adaptive immune response to a subsequent viral vaccine. But that is all published. You can read the paper if you're interested. This is another local cytokine paper where they rightly thought that they would look into IL-13, IL-22, and some mucin genes. And sure enough, they found the IL-13, the red bars are infected animals. They found the IL-13 peak 2 weeks after infection, but not much happening in mucin gene expression. So we don't really know if this is even relevant in Ascaridia galli infections. More to look into as well. But back to the T cell, the important subject. We also, in addition to looking at T cells in the blood, we have been studying in secondary lymphoid organs if we could find any evidence that we saw T cell activation. Using the same method, but mashing up KS patches, fecal tonsil, or Meckel's diverticulum, to see if we could identify lymph blasts as a proxy for recently activated T cells in vivo. And the dark blue bars are Ascaridia galli infected chickens. And sure enough, we definitely in Meckel's diverticulum and KS patches, and to some extent in fecal tonsils, saw an activation of T cells, which made us really happy. Because then there is probably something about T cells that are important in the infection. If we look locally in the jejunum, I also have colleagues who have been trying to identify both lamina propria T cells, but also T cells present in the epithelial layer. And sure enough, this is histology where they look at T cells in the lamina propria, and they see the white bars are infected chickens, definitely increased levels, not massively increased levels, but there is an increased level of both CD4 and CD8 cells in infected chickens. So far so good, but we want more details. And the preferred method is, of course, flow cytometry. And in order to do flow cytometry, you need to dissociate the tissue. And it's really important if you want to study intraepithelial cells and lamina propria cells that you have a method where you get things right. You have sort of a mild procedure to shake out the intraepithelial cells, and then you have a more harsh procedure where you dig deeper into the tissue with some sort of enzyme digestion. And that works beautifully. And there are established mouse protocols, and there's absolutely nothing for chickens. Different labs try different protocols, but there's nothing standardized. And this is something that we're struggling with right now, both to establish the perfect protocol for cell dissociation, but also to find out the different subsets that we expect to see. You need to find antibodies, and reagents for flow cytometry are limited in the chicken, so we cannot stain for everything. But we have to understand what we can combine into the staining panel so that we get the most information out of it. And this is just a poster we're trying to build for the intraepithelial lymphocytes, where we have the gamma delta T cells, the alpha beta T cells, and we also have some non-CD3 positive cells, including the NK cells. And we run into all sorts of problems. For instance, we have a B cell marker that is exclusively expressed on B cells in blood, but all of a sudden we see it all over the place in the intestine. And it really is not nice looking samples. So I just want to finish off showing you an example of a quick and dirty protocol we established, where we used a little bit of EDTA to shake out intraepithelial lymphocytes, and then we stained for various T cell subsets. And as you can see, we end up with a lot of dead cells and debris, but we were able to identify beta T cells, gamma delta T cells, and this CD3 negative population that are probably also including NK cells. And we've got the classical CD8s, classical CD4s, and in the chicken also double positives. And we've got gamma delta T cells that express CD8 and not. And then we've got this additional population. So we are trying to build a nice way of looking at these intestinal cells. And this is just to finish off with very, very recent data. We haven't had time to analyze it. But for instance, we also see this increase in CD4 cells in the jejunum, in the intraepithelial fraction in the Ascaridia galli infected chickens. So we really believe that We need to get these protocols sorted so that we can have much more information about all the cells, basically, locally in the intestine. I think I'll skip that one and then go to this one. So in the coming years, we hope to return to looking at basic immunology, immune responses, also some vaccine development to try to understand what does protective immunity against this pathogen actually looks like. Also the vaccine development is really something that we are keen on, and also discussing with Duranga and Alireza if the extracellular vesicles that the worm actually excretes could be a potential vaccine candidate. But it is by no means easy to develop nematode vaccines. I've listed a number of challenges we have. So we do know that it's not going to be a walk in the park, but we still feel that there is a lot of promise in looking into this research area. And the whole culprit is that parasitic worms, nematodes, are incredibly effective in immunomodulation. The protein products or the extracellular vesicles also contains lipid and RNA, and they are really master regulators of the host immune system. And this is really something we need to understand in detail in order for us to build the perfect vaccine and identify the perfect vaccine candidate. But from next year, we hope to have funding. We think we have funding from the European Partnership on Animal Health and Welfare. And this will allow us to do a little more research within this area. So I'll just finish off by thanking some of the collaborators and thanking you for listening. And I hope you have some good questions for me. Thank you.
Thank you very much, Gina. Do we have any questions from the audience before we move into the next?
Yes, there are actually only, I think, a handful of papers where it has been sort of investigated, but it really is a focal point these years to try to understand how it develops. I remember when I wrote the application 10 years back, we wrote, there are no problems. We do not have any resistance towards these drugs, but later reports actually state that this is not true any longer. So I think we will get more knowledge about it in the coming years. It's not my research field, so probably there is already published more data on it, but they can develop resistance. That's established.
For example, in UK, almost all parasites are resistant to anti-emetic drugs. It's a problem how we treat it.
Exactly. Yeah.
I don't know how we can treat it. Because in Estonia, we place them in a closed farm where we don't even put antibiotics.
Yeah. But that's definitely a fair question.
I think the state and the accident would be the problem people.
Yeah,
Because backyard chicken became so popular, especially during COVID time.
Yes.
What is happening in social media is absolutely terrifying because people, if somebody gets access to Onehealth, for instance, it's so everybody else.
Yeah.
In social media.
Yeah.
And it's absolutely crazy.
Yeah. Yeah. That is true.
Black market for that.
Yeah. And there's also like the welfare discussion that we in Denmark we see, well, the organic farmers are not too concerned about resistance development. They're more concerned about that they cannot label the organic eggs as organic for 5 days after treating. So they tend sometimes not to treat, and then the parasite load gets really high, and it really jeopardizes the welfare of the chickens. So it is really challenging to come up with with good control, that is consumer friendly and animal friendly and farmer friendly. It's yeah, a complex discussion.
No tasty, no problem.
Yeah. Right.
Yeah, get education now, because yeah, it was really presentation. Yeah, my question regarding the model that we spoke about. When you get infection growth or growth, you have more what is the risk and how it happens.
Yeah, we don't have the details. We haven't performed a perfect study. But I think I have a colleague who hypothesizes that it's really also physical constraint, that there is a limitation to how many larvae you can actually harbor at the site in the intestine where the eggs hatch. So you can basically overload the system, and it would never happen in nature. In nature, a chicken would never pick up 1,000 eggs in one dose. It's simply not possible. So I think it's pretty basic that we haven't found maybe the sweet spot of how many eggs can we put into the chicken and get 100% establishment rate. Yeah, we simply don't know where the cutoff is, and we seldom get, or we never get 100% establishment, even when we use 50 eggs. It's yeah, it's not easy.
Another thing regarding the mechanism of this EV release from the market, how it differs from the same type.
Say that again.
Like how it we want to release some vesicles, right? Whereas how it differs from normal cell type, because cancer cell or other.
I don't know.
So in nematode, it seems that there is a lot from the intestine, internally, plus from the skin. So it's a very different. It's not that cellular release that they talk. It's just whole organism like release they always talk about. So there's a lot released from the intestine. So that's why they're interested on using them as immune modulators for maybe vaccine candidates. I don't know, but in humans they have been trying it for some time. But it seems that they are masters of immune evasion. And like
Yes.
Even recently you can see a python nematode in a woman head from Australia. So that means they cross the species and kingdom barriers even little by little, like I don't know, for whatever the reasons.
But they are really masters. It's symbiosis. They are living, or they have developed to really evade the immune
We have missed that earliest communication. That's why maybe you would be interested, like very early. So you see that it happened very early, the quite good resistance, but then after like a couple of weeks back, then you see like again home load high, even though your IgG levels are still comparable with the resistant group, right? So probably something is happening at the
So it really is a balance.
But it's very easy to get the out of the they can culture very easily in the medium. Without I think last week, a couple of weeks back, we had who's the person talk about human health. We had a colleague from the Aarhus Medical School coming.
Peter Nice?
Bradley White.
Look at that one.
Yeah, he came here for the conference actually. So he was showing some data on the system development.
Is that on the website? Oh, I'll have to look at that one. Interesting. But the in vivo situation is really complicated. You have like the EVs and other excretory products from the parasite modulating the immune system fighting back. We also see an impact. This has been more or less published that there is also some modulation of the microbiome, depending whether or not depending on the parasite stages also. So it really is a very complex system to understand in vivo. But the lovely thing, as you say, Saranga, is that the extracellular vesicles are actually quite easy to culture or to obtain.
You can stage wise obtain the like that. Even even I have seen some people get it from eggs as well, and because the egg membrane is also secreting.
Yes.
If there is, there's no any shell or something on the egg, it's just membrane. People have
Yeah, so that's, so all stages are really interesting to look into. It's a question of time and money.
Excuse me, we have question from Zoom audience. So Durmini, please.
Yeah, thank you very much for the interesting talk. I'm just wondering whether there's any, do you find out, or is there any evidence to tell that the co-infection or super-infection with other nematodes together with this galli cause more severe conditions or any sort of things?
Yes, good question, because that's what happens out in production, that usually there's co-infection with other things as well. And there are a few papers where they also tried to establish an experimental infection models with more than one species of roundworms. But what is maybe also interesting is co-infection with bacteria. There's some old data, quite intriguing, stating that if the chickens are infected with E. coli first, it's much more difficult to establish an Ascaridia galli infection. So there are definitely some interactions that we need to study in more detail. And I am by no means expert in this area, but a really good question.
Thank you very much.
Yeah, Subhashis.
You were talking about the ELISA. It seems it was developed in-house, also. Like it looks like a direct ELISA. How was the experience with developing the ELISA? How long it took? How costly it is?
It's very inexpensive.
As long as you have the antibody. As long as you have the antibody.
That is true. And actually the ice type specific antibodies, the price of those have increased. But it was pretty easy to establish really. We spent a little time sort of trying different antigen concentrations but nothing too fancy. But it's not like a fancy ELISA. It's a very crude way of analyzing, and potentially it's not the best antigen. Potentially we're not interested in antibodies towards all antigens present in adult worms, so it really is not the perfect tool to assess more refined questions in relation to the immune response. There are no commercial ELISAs, and back then there was absolutely nothing to compare with. There is a German lab who also developed their own ELISA, but in recent years there are a few publications where they can also look at antibodies in fecal droppings, and there has been but still the commercial interest is not really that great, so no companies have taken up or further validated or refined this particular ELISA. And I think that goes for many of the veterinary pathogens that are not sort of a big deal, that some of the companies are not too keen on developing diagnostic tests for those in terms of serology. Of course, it's easy in a diagnostic setting to develop like a PCR-based detection system. PCR primers are really inexpensive, and that's the molecular diagnostics kits are probably easier to get the companies to develop. But this test was not sort of intended as a diagnostic tool, as a serological diagnostic tool. It was really to evaluate and to quantitate the humoral immune response. Yeah. Good.
Any other questions? Yeah, okay, thank you very much. So we have a small token of appreciation and as a ritual, we are also awarding a certificate. So I have. Thank you very much for the nice lecture.
Thank you very much. Thank you.
It's a photo. Yeah.
Yeah. Thank you. Thank you for listening.